DNA is made of just 4 letters: A, C, G, and T. Biologists often ask a simple question. If we know the current letter, what letter is likely to come next? A Markov chain learns transition probabilities. If we see an A, the next base may be G, C, T, or even another A. In this example, G is the most likely next base. A first-order Markov model only looks at the current base. It doesn't care about earlier history. The next prediction depends only on where we are right now. Different DNA regions have different patterns. Genes follow one set of probabilities. Non-gene regions follow another. By comparing models, scientists can identify where genes are located. In most DNA, C followed by G is relatively uncommon. But in CpG Islands, C is frequently followed by G. Markov chains can learn this pattern and help locate these important regions. Proteins are also biological sequences. Some amino acids tend to appear near specific neighbors. Markov models learn these protein grammar rules. Protein Markov models have many applications. They help classify proteins, identify domains, generate sequences, and study evolution. Sometimes the important states are hidden. We can observe DNA letters, but we cannot directly see exons and introns. A hidden Markov model estimates the hidden biological states. HMM-based tools can predict gene structures directly from raw DNA. They identify coding regions, introns, and gene boundaries. Profile HMMs compare protein sequences against known families. They estimate how likely a sequence belongs to a specific protein group. Markov models can also describe how DNA changes over time. They help scientists estimate mutation rates, study viruses, and build evolutionary trees. For decades, Markov models have been one of the most important tools in bioinformatics. They help us understand genes, proteins, genomes, and evolution. Even in the age of AI, hidden Markov models remain widely used.